Logo for NIH, National Eye Institute. Houmam Araj, Ph.D. Program Officer, National Eye Institute. Lens and Cataract Portfolio Update to the National Advisory Eye Council, January 21, 2016.
Dr. Sieving asked for an update on the Lens and Cataract program. Can you hear me? And Dr. Sieving thought that it might be helpful to have an update for Council as well.
Slide. Outline.
So this is how we can proceed. We'll talk a little bit about the context for the program. Next, we'll talk about some of the themes and success stories, followed by discussion of some of the opportunities and the complexities and challenges that the program faces. And importantly, I think this could be an opportunity for Council to provide feedback both to program and to the Lens community.
Slide. NEI Extramural Program Structure.
Most of the time, by the way, most of the material that I have will be on these success stories from the program, as well as the opportunities that the Lens tissue presents. As you probably already know, the NEI Extramural program has 6 different programs. The Lens and Cataract is one of them. The others being retina, cornea, glaucoma,
strabismus, amblyopia, and
visual processing and low vision.
Slide. Context. FY 2015 budget. Lens and Cataract is about 7% of the $559 million NEI Extramural budget. 2 pie charts. One shows the budget percentage by dollars and the other by grants.
In terms of context, if we look at the fiscal year 2015 budget, the latest budget that we have numbers for, you'll see that the Lens and Cataract program makes up about 7% of this budget. That's whether you measure it by dollar amount or by grants. Within that pie, you'll see that the retina has the biggest chunk,
44% in dollars, 43% in grants,
followed by SAVP,
23% in dollars, 24% in grants,
then cornea,
13% in both dollars and grants,
glaucoma,
10% in both dollars and grants,
and then Lens and Cataract, and finally by low vision,
3% in both dollars and grants. Slide. Lens and Cataract stable and comparable to other DER programs.
I have been the program director since 2006. And for this past decade, the program has maintained the same percentage, that same percentage that I just showed, of the DER budget. Furthermore, if you measure the Lens and Cataract compared to the other programs at DER, you'll see that it's very competitive. It does just as well as any of the other programs within NEI, whether you measure it by the duration that the awarded grants get, or the average priority score that the successful grant, or the success rate. Now, one can think of a number of ways to divide this pie. We can talk about the different funding mechanisms, R01s, R21s, R15s. We can talk about the number of PIs, number of institutions, states, no-cost extensions, or any number of other ways to break this pie. But I suspect that by the end of the day, most of that information would be forgotten. So perhaps we can try something a little bit different. Let's talk about themes.
Slide. Themes. Lens and Cataract research.
For the Lens and Cataract program, actually more generally for Lens and Cataract research, I see 3 main themes: lens development, homeostasis regulation, and oxidative damage. Within each of these main themes, you'll find some sub-themes. So for example, within development, you'll find lens induction, followed by the later stages of lens epithelial cell differentiation into lens fiber cells. You might see me using these acronyms. And then posterior capsular opacification. Within homeostatic regulation, and when it's dysregulated and end up with cataract development, you'll find some significant work being done on the main proteins.
Lens homeostasis, microcirculatory system.
The transmembrane lenticular proteins, aquaporins, and connexins. You also find quite a bit of work being done on the lens ultrastructure.
Cytoskeleton.
Oxidative damage, of course, also can result in cataract development, and the NEI has funded quite a bit of grants over the years in that field. A big chunk of that, I have the crystallines, their function, and their structure. Obviously, it's related. The post-translational protein modifications is also makes up several of the grants funded in the lens and cataract portfolio. Moreover, we have the oxidative damage and the response, the normal responses that the lens has to deal with these insults, environmental insults.
Slide, Lens Induction, 1901. Photos of Hans Spemann, a piece of hair, and illustration of constricted embryo.
Okay, so our first story starts with a Nobel Prize winner and a few strands of hair. You've heard the story of the scientist who was able to use some of these, find his daughter's, actually baby daughter's hair, and be able to constrict a vertebrate embryo and be able to split it into two fully formed embryos. That's the work of Hans Spemann. And what he found was that depending on where you constrict the embryo, you might get viable embryos or not. And so depending on which plane, so if you divide it by this plane, you're not going to get two viable embryos. And so that led him to the idea of a morphogenetic field. That's really the start of it. Next, he moved on to studying more specifically the development of the visual system.
Image of an embryo showing the developing eye cup and lens vesicle.
And what he did was that, this is just an embryo, developing embryo, and what he did is that to take out, this is where the eye primordia, this is where the eye originates, and what he did was take this needle, take this needle, and heat that needle, and just be able to ablate this particular tissue. This is what we know is the optic vesicle. When he does that, the lens no longer develops. He did something else. He actually, this is when ablating the optic vesicle and ending up with no lens. The second thing that he did was to take this covering tissue, this ectoderm, and get some other ectoderm elsewhere from the embryo and just transplant it. And you can see here, the lens actually does develop. Finally, and perhaps most importantly, what he did was to take some of this optic vesicle tissue and transplant it elsewhere. That's where the lens or the eye does not normally develop.
Image shows transplant process, which results in developed lens and eyes.
And yet, believe it or not, he's able to get an eye and a lens to develop.
Slide, Lens Induction, 1901 summary. Image, cross-section of neural tube. Magnified section shows lateral outgrowth of neuroectoderm, forms optic vesicle.
So if you allow me, for the benefit of the audience, just to use this diagram just to summarize the main findings of Spemann. So this is just an embryo in cross-section, and you find the three main layers: the outside ectoderm, the inner, or the middle, I should say, mesoderm, and the most inner endoderm. And so let's just take a closer look at this part, the neural tube, If we look at the neural tube in greater magnification, these are the optic vesicles that we were talking about. And this is the tissue, the ectodermal tissue that gave rise to the lens. And so what Spemann did was, again, ablate this optic vesicle, no lens. You can switch this ectoderm with some other ectoderm coming from elsewhere from within the embryo, and get the lens to still develop, and most importantly, take some of this optic vesicle, this inducing, or what he called the organizer tissue, transplant it elsewhere within the embryo, and still get the lens to develop. All these results led to this really crucial concept of embryonic induction, work that led to Spemann earning the Nobel Prize.
Slide, Lens Induction 2015. Image, ectoderm pathway diagram. Source, Svekel and Asheri Padan.
Now, let's fast forward 100 years or so. And what you find, this is actually coming from a review, a summary from Svekel and Asheri Padan. It's just recently published in Development. And it's really what you see is this symphony of cell determination decision points at each part of this pathway. So if we start out with the ectoderm, and if you activate your BMP, signal transduction pathway, and the Wnt, I know Dr. Vedder is very much interested in this pathway, keep going with this, you'll end up with your epidermis. If, on the other hand, you activate your fibroblast growth factor pathway, follow it with more activation, that's going to lead you to a retina. But most importantly for our purposes, activate FGF, then Wnt, and that will take you onto this pathway for the lens tissue. And you can see some of this perhaps surprises is that you see this link between the pituitary gland and the lens. Moreover, we were hearing today about the olfactory tissue. Well, that's where it's coming from, right? Because you can actually, from this competent cell, if you inhibit the BMP, you'll actually end up with the olfactory placode. Now, keep the BMP on, and inhibit the Wnt pathway, and you'll end up with our lens tissue. Now, there's quite a bit of detail here. And the details really are which pathways are involved, which ones are on and which ones are off. It's not really that relevant, at least for our purposes. So let me just try to summarize what's going on here.
Next slide. Image of lens contact, thickening, invagination, detachment, and differentiation.
And what you find really, this is our optic vesicle again. What you find, this is our pre-lens placode right here, this region. And you find really some fairly simple steps that are involved. The lens placode will just contact the optic vesicle and thicken into an actual lens placode. Then you'll find this invagination within the optic cup. That invagination is followed by a detachment of the lens, and you end up with your lens, followed by the differentiation of the lens fiber cells. You'll find your lens fiber cells here migrating anteriorly. By the way, obviously the light is coming this way. Migrating anteriorly, and the lens epithelial cells just differentiating fully so that later in development they're able to generate more and more of these lens fiber cells. So seems quite straightforward. Contact, thickening, invagination, detachment, differentiation. Yet don't be misled, because underneath these seemingly few steps, there's a lot of stuff going on.
Slide, a cell biologist's dream come true. Diagram showing pathways between anterior neural plate and optic vesicle, preplacodal ectoderm, presumptive lens ectoderm, lens placode, and lens epithelium. Source, Ogino et al. There
is a myriad of transcription factors that are involved. Signal transduction pathways, practically every signal transduction pathway you can think of is involved one way or another. And the second messengers, another zoo of proteins that might be involved, followed by a bunch of cell adhesion molecules and cytoskeleton proteins that are critical for the lens. A lot of this work has been funded over the years by the NEI. Let me just take obviously there's no time to try to explain all of this, but let me just take one part, which is the PAC6. By the way, in terms of development, this is the pre-placode stage, then the presumptive lens ectoderm, then the lens placode stage. So let's just look at this particular stage. Look what PAC6, this is the master regulatory gene. Look how many connections it has with a bunch of different pathways, signaling pathways, FGF, Notch, BMP, and a bunch of different transcription factors. By the way, the PAC6, that's the one that Walter Gehring was able to use and essentially recapitulate what Spemann got, except by simply turning on a transcription factor. It's really amazing what he was able to do. And so he essentially is able to get the eye by simply turning this PAC6 ectopically elsewhere in the embryo. Okay.
Slide, Lens Induction and Development, continuing to yield fruit, last 4 months. Article titled, PROX1 and Fibroblast Growth Factor Receptors Form a Novel Regulatory Loop, Controlling Lens Fiber Differentiation and Gene Expression, by Audette et al. From Development Advance Online Articles. First posted online on December 10, 2015 as 10.1242/dev.127860. Article titled, Connects and Controls Cell Cycle Exit and Cell Differentiation by directly promoting cytosolic localization and degradation of E3 ligase skip2 by Shi et al. From Developmental Cell, Volume 35, 483-496, November 23, 2015. Article titled, Negative and Positive Autoregulation of BMP Expression in Early Eye Development by Huang et al. From Developmental Biology, Volume 407, 2015, 256-264.
Now, lest you think that with 100 years of studies on the lens induction, the field is done, we'll know all the answers. We don't. This is actually just an example of some of the studies, NEI-supported studies, in some fairly high profile publications published just in the last 4 months. So you'll find one on more work on a transcription factor called PROX1. This is from Lakshmi and Lavicu and Duncan's lab. You'll find some more work on the cell cycle regulation. This is coming from Jin Jiang's lab. Interestingly, the part that's doing this regulation is connexin, which is this is the main protein that's what makes up our gap junctions. There's a really cool story here. More work on the BMP, another important signaling pathway. This is coming from the lab of the late Dave Beebe. We miss him.
Next slide. Article titled, Regulation of CMAF and Alpha-A crystalline in Ocular Lens by FGF Signaling by Shia et al. From JBC Papers in Press, published on December 30, 2015 as manuscript, M115.705103. Article titled, CRIM1 Regulates Integrin Signaling in Murine Lens Development by Zhang et al. From Development Advance Online Articles. First posted online on December 17, 2015 as 10.1242/dev/125591. Article titled, RAP1 GTPase is required for mouse lens epithelial maintenance and morphogenesis by Madala et al. From Developmental Biology, 406, 2015, pages 74 through 91.
More work from Renneker and Musil and Sveckel labs. We get some more results on FGF signaling, more on integrin signaling and cell adhesion molecules. This is coming from Mas and Simon John and Richard Lang's lab. And we also get from Vasanta Rao's lab some more really cool results on the GTPases proteins that are critical for cytoskeletal protein regulation. So let me close this first story by this observation. I have yet to see any problem, however complicated, which when you looked at it in the right way, did not become still more complicated. And that couldn't be more true for the lens or really any other field of study. The bottom line is that more research is needed. Okay. So the first story was a story that unfolded over 100 years or so. The second story unfolded over the past year or so. This is the story of the involvement of sterols and crystallins.
Slide. Second success story, crystallins and sterols. Article titled, Lanosterol Reverses Protein Aggregation in Cataracts by Zhao et al. From Nature, 30 July 2015, Volume 523. Article titled, Pharmacological Chaperone for Alpha Crystalline Partially Restores Transparency in Cataract Models by Mackley et al. From Science, 6 November 2015, Volume 350, Issue 6261.
And it actually started out with two different teams that actually started out at very different starting points, and yet they converged and ended up with the same destination. The results were published in the July issue of Nature, and the other team published their results in the November issue of Science. Let me tell you about this group's finding first.
Slide. Forward approach. High throughput screen. Chart shows R120G Cry-AB rapid increase in 500 nanometer light absorption versus flat wild-type Cry-AB as temperature increases.
And so essentially they started out what can be called as a forward approach. And so if you can look at this is the wild-type crystalline alpha B, and it has at these temperatures, it doesn't denature. And yet Dr. Andley, part of the team, had this particular mutation, R120G, that in this crystalline alpha B, the arginine is simply changed into a glycine. And that one change forms this dominant mutation. This is, by the way, coming from the humans. That particular mutation is in the humans. And what Dr. Andley was able to do is to take that mutation and actually knock it into the mouse. And with just one copy, these mice will get cataract. And so this really formed the basis of this study, of this particular approach. Using a high throughput screen, essentially what the team did was to use some of this mutant protein and find, can you add some kind of reagent, some kind of biological reagent, and to be able to shift the profile. This is obviously less stable. Can you make it more stable? Can you make it more in line with the wild type? They screened about 2,500 different molecules and identified several.
Image of aggregated mutant Cry-AB without compound 29 compared to non-aggregated Cry-AB with compound 29, and comparison chart between wild-type Cry-AB and R120G Cry-AB with compound 29.
One of which is what they're calling compound 29. Look what it does. This is untreated. You'll see all this aggregation from the mutant crystalline alpha B. Add some of this compound 29, and the aggregation disappears. All that dissolves, all that aggregation dissolves. And here's a quantification of that. It's an in vitro result, but it shows very clearly, this is the wild type, whether you add just the normally, or you add this compound, no difference, obviously, don't expect one. And yet, in this mutant, this is the mutant without anything. Add the compound 29, and before you know it, you have a lot more that is dissolved than without this compound 29.
Image of compound 29 molecular structure.
This compound 29, when they looked at what it was, turned out to be very similar to Lanosterol or Lanosterol, depending on how you want to call it.
Chart shows treatment results of wild-type mice with compound over a period of 2 weeks.
Most crucially, the team followed by obtaining in vivo results. And so remember the mice that I told you about? These are knock-in mice having this mutation. These are just wild type. Obviously, wild type mice, you don't expect to have them a lot of cataract, right? And yet with these knock-in mice, if you don't do anything, you'll see quite a bit of cataract, right? By the way, these are just two different groups. One young animals and one a little bit older animals. And this is what they were treated with. When you treat with this particular concentration at this number of weeks, you'll get a significant reduction in cataract development, in opacity, really, lens opacity in these mice. Do that for the older group of animals, and sure enough, you get that. So it's really fascinating to get this in vitro evidence followed by some very specific in vivo evidence of the efficacy of this particular compound to reverse cataract development.
Image. Compound 29 binds a groove between the Cry-AB protomers.
As if that's not enough, the team goes ahead and does some more molecular modeling experiments and finds that this particular compound fits in a really cool groove between two dimers of the alpha B. So essentially, it makes sense why it could potentially stabilize this interaction between the alpha B dimers.
Slide. Reverse approach. Mendelian genetics. Inheritance maps showing affected siblings in a consanguineous family.
Let's move on to the second team. The second team has pretty much used the reverse approach. Started out with a consanguineous family with 3 members affected and cloned the gene involved. Found out that it actually was involved in, it affects this particular enzyme.
Molecular model of Lanosterol synthase.
Lanosterol synthase. More importantly, it's actually turned out the mutation that they're studying turns out to be in the catalytic domain of this particular enzyme. So it all makes sense.
Image shows control plus cholesterol and plus Lanosterol data over 900 minutes. Aggregation decreases with Lanosterol.
Of course, you expect to see some in vitro data to show, and so that's obviously pointed investigators to the importance of Lanosterol, given the importance of this enzyme in this mutation. So what they do is to simply, these are just some cells, some human lens progenitor cells, expressing a mutated alpha A crystalline, okay, this particular mutation. And what they do is that if you, this is the control, you see you'll see your condensed, your aggregated alpha A crystalline, the mutant, because it's a mutant. And you add cholesterol, no difference really, it's pretty much the same. Yet, look what happens when you add Lanosterol. Over time, it actually diminishes this condensation, this precipitation of protein, until at later time points, you actually don't have any more precipitated mutant crystalline anymore.
Image of dog eye before and after 6-week treatment, 3 times daily, along with chart showing cataract stages before and after treatment.
As you'd expect with a Nature publication, they wanted to get in vivo evidence, and sure enough, except then they used the canine model. And so what they do is test some dogs with cataract, and then just pretty much do this topical treatment 3 times daily of adding Lanosterol. That's all they did. And then test for the cataract score and find out that it is significantly diminished. So it's really cool work. Again, has the whole story from A to Z. Now, Lanosterol. Obviously, that came as unexpected.
Slide, Lanosterol. Who ordered that? Diagram of cholesterol biosynthesis pathways.
Why Lanosterol? Now, Lanosterol obviously is an important part in the cholesterol biosynthesis pathway. Obviously, not going to go through all that. So you can see starting out with fatty acids, it goes a bunch of different steps. You'll get from this particular, this compound, from this particular compound, you get your, the Lanosterol synthase is able to synthesize Lanosterol and goes on to cholesterol. Now, there's one more important part of this pathway, which is really this pathway. Let me show it to you in magnification.
Magnified HMG-CoA pathway.
This particular enzyme, the HMG coenzyme A reductase, is critical because that's the one that statins inhibit. If you're going to reduce cholesterol, that's the enzyme that this statin, that is obviously very commonly used, that's what it targets. And so that obviously opens up the question of the involvement of statins and cataract. And you'll see actually this fairly recent publication.
Article titled Statins and Cataracts, A Visual Insight by Jean M. Dobrzinski and John B. Costas from Current Atherosclerosis Reports, 2015, 17:477. Quote, the data on the relationship of statin therapy to the development of cataract are not consistent.
They actually try to make sense of the different data. The bottom line is the conclusion. The data on the relationship is really not consistent one way or the other. And as you can imagine, more studies are needed.
Slide, protein misfolding diseases. Table 2.1, representative protein folding diseases. Table data listed in this order: disease, protein, and site of folding. Hypercholesterolemia, low-density lipoprotein receptor, ER. Cystic fibrosis transmembrane regulator, ER. Phenylketonuria, phenylalanine hydroxylase, cytosol. Huntington's disease, Huntington, cytosol. Marfan syndrome, fibrillin, ER. Osteogenesis imperfecta, procollagen, ER. Sickle cell anemia, hemoglobin, cytosol. Alpha-1 antitrypsin deficiency, alpha-1 antitrypsin, ER. Tay-Sachs disease, beta-hexosaminidase, ER. Scurvy, collagen, ER. Alzheimer's disease, beta-amyloid presenilin, ER. Parkinson's disease, alpha-synuclein, Cytosol, scrappy Creutzfeldt-Jakob disease, prion protein, ER. Familial amyloidosis transthyretin lysozyme, ER. Retinitis pigmentosa rhodospin, ER. Cataracts, crystallins, cytosol. Cancer, P53 cytosol. Source, Dobson, 2001.
Now, it's as important as it is the angle of the Lanosterol and the statins. Potentially even more important is its involvement in protein misfolding diseases. Now, cataract really is an example of a whole bunch of different proteins that are really what's involved is these proteins that somehow aggregate. And you see some fairly well-known diseases, Alzheimer's and Parkinson, CF disease. And so that opens up the question, since at least in this model, in this cataract model, a particular compound is able to reverse this protein aggregation, could it be that maybe some of these other proteins can also be reversed? That could be huge.
Slide. Why should we care?
So I told you kind of a little bit about the context, told you a couple of stories about some success stories from the lens field, but why should we care? Let's try to address that more directly.
Slide. Opportunities. Mouse lens tissue from embryonic day 18.3 through 24 months.
I would say in a word, it's the opportunities that the lens tissue, the lens system provides. There's a beautiful story behind this from Steve's work, but I don't have the time for it.
Slide. Beauty of the lens system. Simplicity. Cross-section schematic of the lens. Source. Kodarami. Image showing lens elimination system. Source. Bassnet.
So let me just move on. Beauty of the lens system. I think the main thing, the thing that just jumps at you is the simplicity of the system. Two cell types, just some lens epithelial cells and some fiber cells. Two cell types. Couldn't be any more simple. These lens epithelial cells that develop and grow into about lens fiber cells that probably are 100 times longer than these epithelial cells. Of this protein in these lens, specifically the lens fiber cells, 35% of it is a protein. This is the highest level of protein anywhere in the body. And you can imagine why that's needed, because if you're going to get refractive power, you need to have a lot of protein to do that. Lens is able to do that. Moreover, since the nuclei, or really any organelle, cell organelle, if it comes in the path of the light, it can diffract that light. So evolution decides to get rid of nuclei, ribosomes, and mitochondria. Let's look at some of this diagram. You can see it. These are the lens. By the way, we're looking right here pretty much at the equatorial region. And these are the lens epithelial cells. They're maturing into lens fiber cells. And as they're going closer to the lens nucleus, you'll see the nuclei being eliminated, as well as the mitochondria. So that by the time you get to the center of the lens, you essentially get this organelle-free zone. So let's think about that for a second. So you have a tissue that has no nuclei, meaning no lens transcription, no mRNA transcription, no ribosomes, which means no protein translation, no mitochondria, another problem, and yet that tissue is somehow able to do all of its normal functions. How is that possible? Now, I know some of you might be thinking, well, red blood cells also don't have nuclei, except red blood cells are eliminated within a few months' time. These cells have to stay with us for the rest of our lives. That's really a challenge. On top of it, as if that challenge is not enough, the lens has to do it with no blood vessels. And somehow evolution solved that problem.
Slide. Beauty of the lens system. Complexity. List of crystallins / filaments and total post-translational modifications. Source. Srivastava.
Okay, well, some of you might think, well, simplicity is not my thing. I want to see some more complexity. And I would say the lens has a number of different examples to express that. I'll just settle for this one, which is the post-translational modifications that I mentioned before. If you look at, these are, by the way, some species of crystallins. And look at the different modifications that you can see. These are the different locations within the different protein that you find this particular modification. Some of these are kind of expected. Oxidation, deamidation, methylation. Well, yeah, I expect. Some other ones are, I'm not even sure how I can pronounce this. So you find these common post-translational modifications and rare ones. Moreover, this list actually is partial one in that it doesn't even say about the truncations that take place into these crystallines. So you'll find certain domains, usually probably the carboxyl domain, that somehow it gets chopped off at different locations within the protein. And somehow the protein sometimes still stays functioning and others not.
16 crystallines.
So crystallines, there are a bunch of different species. This only has pretty much half of the other ones that are available. Moreover, these modifications are not just for crystallines. You'll see them for connexins and aquaporins, and a whole bunch of different species of these. As if that's not complex enough. It's very difficult to tell. How can one tell whether that particular modification is pathogenic or ends up being just a normal part of aging? Moreover, how can you tell if this is cause of the pathogenesis that you see versus a result of the pathogenesis? So it's really a complicated system.
Slide, clinical significance. Pie chart shows that cataract and uncorrected refractive errors are the leading causes of avoidable visual impairment with 33 and 42%.
Some might say, well, yes, for the basic science, we want to see some more clinical significance. And so for a clinical significance, if you look at, this is just the global burden of disease, and you'll find that cataract forms about 33% of the visual impairment. Now, but that's just visual impairment. What about blindness?
Pie chart showing unoperated cataract is the leading cause of avoidable blindness with 51%. Source, Pascalini and Mariatti, 2012.
For blindness, actually, the significance goes up. You end up with fully half of the burden of blindness is from disease. 20 million people. How do you make that number count? It's very difficult. We go to numbers to make sense of these numbers. Difficult problem.
Slide, posterior capsular opacification. Diagram showing intraocular lens with capsular wrinkling. Source, Warmstone.
Okay. From posterior, I see I'm running a little bit out of time. So posterior capsular opacification. I would say that that's, if you think that, well, we can replace the lens. Yes, we can. So the global problem is not really our problem. It's true. But posterior capsular opacification is.
Schematic showing that increase in TGF-beta signaling leads to increase in myofibroblasts and lens epithelium during cataract development.
And I'll just say that we have a nice system that is that is funded by the NEI to deal with this problem. I'm going to skip these.
Slide, opportunities related to AGI, lens in axon guidance. Article titled Lens-Derived Semaphorin 3A Regulates Sensory Innervation of the Cornea by Peter Weiler-Wigal and Marianne Bronner-Fraser from Developmental Biology. Article titled Nerve Repulsion by the Lens and Cornea During Cornea Innervation is Dependent on Roboslit Signaling and Diminishes with Neuron Age by Schwend et al. Images showing nerve repulsions.
Which is what I wanted to do, was to show the link of the lens that it actually can link up with the Audacious Goals Initiative. Both the lens, believe it or not, has a role in axon guidance and axon regeneration.
Slide, more things that make you go,
Okay, so let me just kind of wrap this part up and say, to be surprised, to wonder, is to begin to understand.
Jose Ortega Y. Gasset.
A bunch of other things that really makes you wonder. Lens, no lipid turnover in the lens for the entire lifespan of the human.
Hughes et al., eLife, 2015, 4:E06003.
Why is it that the lens never develops cataracts?
Does anybody care that the crystalline lens never gets cancer? Clinical and Experimental Ophthalmology, 2013.
Questions that are really begging for question of mechanism, how?
Article titled Crystalline Eyes of Chitons Inspire Materials Scientists from Science, sciencemag.org, 20 November 2015.
Latest issue of Science, a very late issue of Science, it talks about this really cool crystalline in a mollusk.
Article titled Absence of Spark Leads to Impaired Lens Circulation by Grieling et al. Experimental Eye Research, 2009 September, 89:3:416-425.
And this is just for Dr. Ray. I know he's interested in spark, so I just thought throw that in.
Slide, opportunities at NEI.
More lens opportunities. I'll keep that pretty much talked about what I wanted to say, which is, remember, now the crystallines are known for the refractive function and for the chaperone function. Fine. But what about the other non-refractive and non-chaperone functions? We know close to nothing about them. Connexins. Connexins are obviously, that's what makes up our gap junctions. What about the non-gap junctions? We just saw a hint of that with Dr. Jiang's work that I just mentioned a while ago. Aquaporins, yes, water channels. But what about the non-water channel function of aquaporins? Not only within the lens, you can also talk about opportunities within the eye. Both, the crystallines are obviously critical for the RPEs and the RGCs. Aquaporins, cornea, trabecular meshwork.
Slide, opportunities at NIH.
Finally, this is last, the one before last. In terms of opportunities at NIH, I kind of just thought it'd be fun just to kind of list some of the different opportunities that can link up with the lens can contribute to. Obviously, for NIGMS, they're very interested in chaperones. By the way, we are involved in this cryo-EM initiative as well as the glycoscience initiative. This really could be beneficial to the lens community. NHGRI, the age-related cataract development. NINDS, I talked about the protein aggregation diseases. NHLBI, if you think that the connexins are important in the lens, wait until you see what they do in the heart.
NIDDK.
Similarly, aquaporins and the kidney.
Diabetic cataract. NIAMS: fibrosis and wound healing. NIA: senescence. NCI: only tissue that does not form tumors, parentheses, yet epi origin. NIBIB: imaging modalities. NIEHS: environmental damage, smoking. NIMHD: cataract more prevalent in African Americans. NCCIH: cataract and dietary supplements, parentheses, antioxidants.
Let me just move on.
Slide, complexities and challenges.
So, complexities, obviously, I kind of showed you the opportunities. There are obviously complexities, as with any system. Biology, at the end of the day, the cataract is an aging disease, and that's not an easy problem to deal with. There's the issue of how do you deal with the more complicated, the age-related cataract mutations. Most of the ones that we've been found so far are Mendelian forms. How do you get to the other ones? Model system limitations.
Different species, different systems, parentheses, in vitro and in vivo.
Also, reproducibility in any of these studies, really. And there's a cool editorial by Dr. Zarbin that you might want to look at.
The elusive nature of truth in scientific studies and the importance of peer review, TVST editorial. Slide, further complexities.
Silos and fragmentation.
Molecule de jour versus systematic approach. Limited resources and competing demands. Paucity of new investigators. ARVO abstract data and intramural pipeline. Retirements that are not replaced. Philosophical: laissez-faire, proactive versus hybrid. Inertia and consensus. Unintended consequences. To change and change for the better are two different things. German proverb.
I'll just skip that. And again, a bunch of other problems that I think are very similar to other fields as well. This is, I would say, crucial for the lens field in terms of just really needing to acquire and attract more young investigators. I said this is the last slide, but I truly now have just one more. How do you advance that field? How do you help the community? Do you try to do something kind of more hands-on or more laid back, and watch out for the unintended consequences and the potential for doing more harm than good?
Next slide. Book, Second Sons, by David Oliver Reelan. Quote, Second Sons should be required reading for anybody with an interest in humanitarian philanthropy, or, for that matter, a desire to feel a little better about the world. Outside Magazine.
Finally, I'll close by telling you about this book. I usually hesitate to recommend a book because we all have different tastes, but I think in this case, an exception is warranted. Read about the story of the Himalayan cataract project, told by a master storyteller, whom we unfortunately lost to suicide before his book came out. I think you'll find it worth your time.
Slide, NAEC feedback. to program ? To Lens Research Community ?
And I thank you for your time.
Logo for NIH, National Eye Institute.